What's up, guys? Back with another educational video, and this week doing a video that I probably should have done a long time ago. We're talking about different types of studies and their limitations and what they're good for. I wanted to do this because what I see a lot of is, one, people get really confused because they say, well, this, one study says this and another study says this, and I don't know what to believe. And then also, when a study comes out, invariably you have one group of people who agree with the study, who prop it up as the greatest thing ever and there's nothing wrong with it. And then you have another group of people who nitpick every little thing, every little limitation of a study. Okay, so first off, you have to understand that all studies have limitations. You cannot create a study that answers all the problems of the universe in one study. Every study has limitations. And I think it's important to go through and understand a few different basic types of studies. One of the first ones, and kind of the lowest form of evidence, is, for example, case reports or expert opinions. So a case report is where usually it's an isolated individual, there's no control group, it's just this person, and they're studying the effects of something. So this happens a lot, for example, in bodybuilding contest preps. So there's quite a few case studies on natural bodybuilding competitors. And the reason there's case studies on them is because most of them don't want to be involved in, say, a randomized controlled trial because they don't want to give up autonomy over their diet. So for example, if you were to put bodybuilders into a randomized controlled trial, let's say one group was getting high protein, the other group was getting low protein. As soon as you randomized bodybuilders to the low protein group, they're all going to quit the study. What you're doing is you're saying, okay, we can't really randomize in this situation, so we're going to look at one person and observe the effects. Now, what are the limitations of this? Well, limitations of this is you can't really infer causality and you only have one data point. You're just kind of observing this. So it's useful to gather ideas, but you can't really draw many conclusions from it. The next sort of thing would be like cross-sectional epidemiology. So this is where they look at a population and like a trait or a disease or something like that and try to make associations. So for example, they may look at something like heart disease and then something like, for example, meat intake. And we hear this a lot. Okay, well, high meat intake is associated with heart disease. And so that may be something where they survey 200,000 people. And they're looking at, okay, how many of these people have had some instance of a cardiovascular disease incident versus who eats different levels of meat. And then they look at, okay, what things correlate. And usually they're looking at more than one thing. So they're not just looking at meat. They might be looking at like vegetable intake. They might be looking at total calories, whatever, what have you. So what's the advantage to this type of research? Well, the advantage to this type of research is it doesn't take a real long time to do because it's just kind of questionnaire based for the most part. And two, you can get a really large amount of subjects. So you get a large amount of human subjects. I mean, we have epidemiology that can go up into the millions in terms of number of people. So you get a really large subject number. What's the downside? Well, the downside is you can't really infer causation from that because you're just looking, one, you're looking at a point in time, and two, there's what's called confounding variables. So let's take meat, for example, because this is a popular one. I've talked about it before. People who eat higher levels of meat also eat higher calories. They tend to eat less fruits and vegetables. They tend to be more likely to smoke, less likely to exercise. They tend to live a more unhealthy lifestyle because, especially in Western society, people who eat a lot of meat also tend to eat a lot of processed meat, a lot more junk food, those sorts of things. So it's really hard to pick apart and covariate all those confounding variables. That doesn't make epidemiology worthless. We just have to understand the limitations. We're looking at correlations, but we can't infer causation from that. But it can give us ideas about things to examine further. You can start to be a little bit more confident in epidemiology when there is repeated associations that are very close across different populations over long periods of time. So that brings me to my next sort of type of study, which is cohort studies. Now, a cohort is where they take a certain group of people and they monitor them for a period of time, and then they attempt to sort out based on different variables. So it's kind of like a cross-sectional study, except done over a period of time. So they might look at, okay, in this cohort of 10,000 people, let's see what happens over 10 years from people who eat less meat to people who eat more meat, for example. Now, what's the advantage of this versus a cross-sectional epidemiology study? Well, the advantage of this is you're doing it over a period of time. You're not just taking a point in time and looking at it. What's the disadvantage? Well, the disadvantage is you're going to get a little bit less people than you would for a cross-sectional epidemiology study because you have to follow them over time. So it's hard to get people to, even like for a phone call, for a study, like for 30 minutes. So it's hard to get them to do it, every year, and you have attrition rates and those sorts of things. The other downside is it still really is correlation, and it's hard to infer causation. Now, once again, there are certain things you can covariate out. For example, there was a recent study where they looked at meat intake, but they also looked at it with different levels of fruit and vegetable intake. And what they found was that, this was with cancer, they found that at the highest level of fruit and vegetable intake, it didn't matter if you ate high meat intake, you still had a pretty low relative risk of developing cancer. What we can possibly infer from that is perhaps the association of meat with cancer, or specifically red meat with cancer, is more so to do with the fact that people who eat red meat tend to eat less fruits and vegetables, and if they eat more fruits and vegetables, that relative risk goes away, or at least is reduced. Now, the next tier up of evidence would be things like randomized control trials. What is a randomized control trial? Well, ideally in a randomized control trial, you're comparing at least two groups. You are controlling quite a few variables, and hopefully you're only having like one variable that you're trying to change. There's different types of RCTs. There's, inpatient, there's free living, those sorts of things. But the advantage to a randomized RCT is, one, by randomizing, you can get rid of some of that bias that you see in epidemiology. So for example, if you give the instructions of one group is going to eat 8 servings of meat per week, while the other one is going to eat 4 servings of meat per week, but we're going to have them eat the same number of fruits and vegetables and the same number of calories, and we are going to completely randomize them just based on their body weight, for example. When you do that, you get rid of some of these confounding variables because, you could say, well, what if one group exercised more than the other? Well, through the process of randomization, it's unlikely that that's going to happen because there will be some people who exercise more than others. If that's not an exclusion criteria, you would think that distribution would be pretty consistent because they've been randomized. That's one of the big benefits. Now, people will say, well, that sounds great. Why don't we do more randomized controlled trials? Well, randomized controlled trials have limitations as well. So, for example, it's hard to do really long-term randomized controlled trials, especially ones that are highly controlled. So if you take, for example, a metabolic ward study, which is kind of the highest level of nutritional randomized controlled trial, where they literally house people for weeks. You can control everything. You control their calorie intake, protein, carbs, fats. Literally, they're only eating the foods you give them. People say, well, that sounds great. Well, the problem is they're extremely expensive. You have to pay people because they're basically in food jail, right? So most of these studies, you have to pay each subject thousands of dollars because they can't go do their jobs. They just have to live there for a period of time. So you might only have 6 to 8 subjects in a group unless it's a crossover design. Whereas with a large randomized controlled trial, you could do, say, 1,000 people. But now, if it's going to be any period of time at all, it's going to have to be relatively free living. And as we know, people are notoriously bad at being adherent to diets. If you want to have in a randomized controlled trial, if you want to have high subject number and very controlled, it's going to have to be an animal study because otherwise it's just going to cost too much money to run a long-term, tightly controlled randomized controlled trial. That's just the facts. Even something like a 4-week inpatient metabolic ward study with, say, 20 subjects would probably run you, depending on how many measures you're taking, anywhere from $500,000 to $1 million. I'm just guessing, but I think that that's probably pretty darn close. So it's very difficult to do these highly controlled, long-term randomized controlled trials. Further, a randomized controlled trial isn't always the right option. Now let me give you an example as to why. So I was in a debate on Twitter where somebody brought up that, you can't really say that, where somebody brought up that LDL cholesterol hypothesis is garbage because if you look at the randomized control trials, they don't really show much. It's all epidemiology, et cetera, et cetera. Well, the problem with something like examining LDL and heart disease is when you're looking at heart disease, that is a lifetime exposure risk disease, meaning that for it to make a difference, for you to see differences, you're talking about your exposure to various levels of LDL from literally when you're born to when you die. If you do a 1-year randomized control trial and you don't find a difference in cardiovascular events, well, it's just 1 year. You might take people and randomize them to high saturated fat versus low saturated fat and not see a difference. But again, it's just 1 year when you're trying to compare a lifetime of risk. So in particular, for that sort of measure, something like cohort studies may actually be better because you can examine more long-term risk. Now, I think something like Mendelian randomization is actually best for that. Now, what Mendelian randomization is, they take people and look at their genetic code, for lack of a better term, and find people who genetically, based on polymorphisms in their genes, secrete higher or lower levels of various things, or have higher or lower concentrations of different receptors or enzymes or what have you. So a great example of this is there are high and low secretors of LDL cholesterol. And you can do Mendelian randomization with these groups where you look at people over the course of time who they have a polymorphism on their gene that either causes really low cholesterol or higher than normal cholesterol, and see, okay, who has more cardiovascular events over time. And in those Mendelian randomization studies, it's pretty consistent that the people who have higher LDL have higher incidence of cardiovascular disease. And that holds true even when you stratify for HDL and inflammation. So for example, one of the criticisms of the LDL cholesterol model of cardiovascular disease that people will say is, well, if you look at people who have high HDL, low triglycerides, and relatively high LDL, they still have a pretty low overall risk. And that might be true. But when you equate all those variables through Mendelian randomization, you still see people who have high HDL, low TAGs, and low LDL have a lower risk than people who have high HDL, low TAGs, and high LDL. Meaning you can still be relatively low risk if you have high LDL if you're otherwise healthy, but you're never going to be as low risk as somebody who also has all those things plus low LDL. And we see that too with things like inflammation. So at high and low levels of inflammation, the people who have high and low levels of LDL, in each situation, the people with lower LDL have lower risk of heart disease. So again, that's a situation where you have to know what you want to examine and what study is most appropriate for that. And then the highest level of evidence we have are things called systematic reviews and meta-analysis. So these are basically a study of studies. So you're going to have inclusion criteria. So for example, you might want to look at, say, LDL cholesterol or saturated fat in heart disease, right? So you say, okay, we're going to include any study where they had a difference in saturated fat intake of, let's just say 10% of dietary calories. And they examined them for at least 5 years for different cardiovascular disease risk things. Okay, so that could be an example of various inclusion criteria. And your inclusion criteria are really what determine the quality of a systematic review. And this is something that you have to look closely at, because just because something's a systematic review doesn't mean it's always good. But these sorts of reviews, based on their inclusion criteria, attempt to basically synthesize the studies that fit that inclusion criteria and come up with an overall consensus of that data. So these meta-analyses, you can do meta-analyses of epidemiology, and you can also do meta-analyses of randomized control trials. And so the advantage you have is now you're taking something that, especially for RCTs, which might have low subject number, you can really increase the number of subjects because you're combining studies that are similar. So meta-analyses, systematic reviews, when we're looking for the consensus of the data, that's where we should start. Now, what I always tell people is if the meta-analyses and systematic reviews, the randomized control trials, and the epidemiology all line up, then you can be pretty confident that whatever effect you're looking at is probably pretty likely correct, that the data is pretty consistent. But if you have disagreement between meta-analyses or the data seems equivocal, then I kind of default to, all right, what do the most tightly controlled studies show? Then if those studies are still equivocal, my personal opinion would be, okay, well, we just don't know. Now, one other type of evidence I really haven't covered is what's called mechanistic data. And this is usually done in vitro, which is cell culture, or in animals, because you can kill an animal, dissect it fully, look at all the tissues, those sorts of things. You can also do stuff to animals that you can't do to humans. This is why we have a lot more highly controlled data in animals. Now, what's the limitation on animals? Well, the limitation on animals is they aren't people. The other limitation with mechanistic data is it does not always pan out. With mechanistic data, and let's take, for example, something like mTOR and cancer. Now, there's been a big push to kind of make mTOR as the bad guy for cancer. All right, so mTOR is a target of a cancer therapy drug called rapamycin. It inhibits mTOR. This seems to slow tumor progression. Okay, well, people have taken that and said, well, we really should be trying to limit mTOR activation. Well, what activates mTOR? High quality protein sources that are high in leucine, because leucine activates mTOR. And people have taken this and say, okay, that means leucine is going to give you cancer. Again, you're not just dealing with an isolated variable. Everything exists in concert in metabolism. If, for example, mTOR was an independent risk factor for developing cancer, not only should leucine and high protein diets cause cancer, resistance training should cause cancer as well, because resistance training activates mTOR far more than leucine or protein does. But we don't see that. We see people who resistance train actually have a lower risk of cancer. So this means that even though that mechanistic data, it seems to kind of set up to tell us something, when we try to translate it into a whole human, it doesn't seem to hold up. Now, if you have epidemiology, mechanistic data, randomized control trials, and systematic reviews and meta-analyses all telling you virtually the same thing, then you can be really, really confident that what you're seeing is legit. But always beware of cherry picking. So let me give you one more example of that. We know that smoking causes lung cancer. We have mechanistic data. We have case control studies. We have cohorts. We have large epidemiology. We have systematic reviews and meta-analyses. But even if you look at those systematic reviews and meta-analyses and you look at a forest plot, you can see random studies where smoking doesn't have any effect on cancer or even has a slightly protective effect. Guys, one thing to remember is you can always find a single study to support whatever your belief system is. That's why it's really important not to get hung up on single studies and look at what the overall body of evidence is telling us. And that's why it's also really important to follow people who know how to interpret this stuff and who don't cherry pick. Unfortunately, it can be very difficult to do that because everyone wants to be an expert. But one of the things I'll always say when you're trying to find people to follow who are real experts is usually real experts will not make really big, bold claims. They won't cherry pick, and they will say the following magic words: I don't know. If you're following someone and they always have an answer for a question and they never say I don't know, then they're probably not an expert. Guys, I hope this video helped you understand research a little bit better. If you liked the video, please click like and subscribe. Please visit the links in the description. Check out some of our products, including our nutrition coaching app, Carbon, which is killing it, as well as our educational books and ebooks. I hope you guys have a great week, and I'll catch you next week.